Okay, so we have here the different virulence factors utilized by the microorganism in order for that to prevent here the killing of the host immune system. So the first one we have here the able to resist the phagocytosis basically because of the capsule. So the capsule here serve as the protective coating that will try to mask here the bacterial antigen, and therefore your phagocyte includes your PMN, polymorphon, nuclear neutrophil, and we have also here your macrophages. So therefore, the phagocytic system could not identify the presence of your antigen because this being masked here by the presence of your capsule. So at the same time, that one could also prevent also here the activity of the complement that helps our body to eliminate the pathogen in our body because of that protective coating of your bacteria. In the case of your Staphylococcus aureus, it has this protein A. Okay, the protein A of your Staphylococcus aureus try to block here the binding of the Fc portion of your IgG, which is your antibody. Antibody help in our body para mag-eliminate ng pathogen. So, if this one is your antibody, ito yung Fc portion. So, this Fc portion we're able to bind here to your bacteria normally, usually. So, pag nag-bind dito ang antibody natin, your phagocyte could recognize that that one is a pathogen if may naka-coat sa kanya na antibody. And that's the process, you call it one as your opsonization. In the case of your Staphylococcus aureus, because the protein A, it tried to block here the synthesis or the binding of the Fc portion of your IgG antibody, and therefore your IgG antibody could not coat your Staphylococcus aureus, and therefore preventing here the recognition of your pathogen by your phagocyte, and therefore could not be eliminated as well by our phagocyte. Your capsule here may be in the form of your polysaccharide capsule in the case of your Haemophilus influenzae. We have also here your strep pneumoniae. We have also your Klebsiella pneumoniae. On the other hand, your Pseudomonas aeruginosa would have here the slime layer. So remember that your slime layer and capsule belongs to your glycocalyx, only that they just differ in such a way that your capsule is organized, and that one is firmly attached to the cell membrane. Your slime layer is disorganized, and that one is loosely bound to your cell membrane. Escherichia coli would have also here the capsule in the form of the K antigen, and your Salmonella typhi here would have your capsule in the form of the VI antigen. So this capsule here try to prevent the phagocytosis. So they're able to resist the activity of your phagocytosis because here again this being masked by the protective coating of your capsule. On the other hand, we have also here the mechanism that try to kill your phagocyte. Yung kanina, naiiwasan nila ang phagocyte, ito naman, papatayin na talaga nila si phagocyte. Example for that, we have your streptococcus. So, meron siyang hemolysin. So, hemolysin here try to lyse your RBC. And at the same time, that one would have also a toxic activity with your WBC. Your staphylococcus on the other hand, meron siyang leukocidin. So from the words very specific for the leukocyte lysis. Leukocidin, the form of your Panton Valentine leukocidin. The second mechanism here that try to serve as the virulence factor of your bacteria to cause here the disease is primarily because of its ability to attach to your host tissue or cell by your adhesive proteins in the form of your adhesins. So example for that, we have your fimbriae. So fimbriae here is not for the motility. So that one would allow here the attachment of your bacteria to your host tissues. Example of the fimbriae, we have here your Neisseria gonorrhoeae. We try to use the fimbriae in order for that to attach to your urogenitals that try to cause here your gonorrhea. Another one, your ETEC, your enterotoxigenic E. coli strain. Okay, responsible here for the traveler's diarrhea. Di ba ang Giardia natin is traveler's diarrhea na parasite? Ito ay traveler's diarrhea na bacteria. ETEC. Okay, that's your enterotoxigenic E. coli, also used as the fembri, in order for that to attach to your intestinal tract. That would result here to your diarrhea or enteritis manifestation. Okay, the next virulence factor will be the ability of your microorganism to survive even inside your phagocyte, even after your phagocytosis. Okay, so we have the term. So if this one's your phagocyte, okay, the phagocyte contains the vacuole. Pag ang microorganism natin in-engulf, kinain ni phagocyte, your microorganism bacteria try to go inside the vacuole. Pag nasa loob na ni vacuole ang ating bacteria, you call it your phagosome. Another one, your phagocyte, meron siyang mga granules dito. That's your lysosomes. So the lysosome here contains the granules containing the enzyme, like your myeloperoxidase, and that one is responsible here for the intracellular killing or digestion of your bacteria. Okay, pag nag-combine ang phagosome sa ating lysosome, you call it your phagolysosome. So we have here the mechanism here how the bacteria were able to survive intracellular even if they are already engulfed by our phagocyte. The first one, so those are the bacteria which try to prevent the fusion of your phagosome and your lysosome. So pag hindi siya mag-fuse, hindi mag-release ang content ng lysosome natin which are your enzymes and therefore hindi siya mapatay. Second one, okay, even if they try to fuse here ang ating phagosome plus your lysosome, your bacteria try to inhibit the activity of your lysosomal enzyme. So therefore, it try to prevent here the killing pa din. Third one, some of your bacteria try to escape your phagosome. So inside the phagosome, pumunta dito sa cytoplasm. Pag nasa cytoplasm ang ating bacteria hindi siya mapatay. And therefore able to survive here intracellularly. On the other hand, the host also here would have the following substance, lactoferrin and your lysozyme, which would have here both the antibacterial property. However, in the case of your Neisseria meningitidis, it try to utilize your lactoferrin as its source of its iron for its growth. So, instead na dapat papatayin siya ni lactoferrin, ginamit niya sa lactoferrin para mabuhay siya. Napakautak niya. And therefore, hindi siya mapapatay. So, we have here the different bacteria that able to survive intracellular within your macrophages. Mycoplasma, Mycobacteria, Chlamydia, and we have also your Listeria. Okay, the last one, we have here your mechanism here that try to serve as a virulence factor. So, some bacteria try to release here the toxins, which are poisonous substances which try to interact with your host that try to cause here damage to your host. So, we have your exotoxin and we have also your endotoxin. Okay, then we have here the difference between your exotoxin versus your endotoxin. So exotoxin is primarily produced here by both gram-positive and then gram-negative organisms. So this is toxin that's being released extracellular, kaya nga exotoxin. This is not built inside the body of your bacteria. But rather, one is a toxin that's being released by your bacteria to the extracellular environment. So your exotoxin is made up of two subunits. One unit is the non-toxic. The non-toxic subunit responsible here for allowing the exotoxin to bind to your host tissue. And the toxic unit on the other hand responsible here for the toxic effect. And the toxic unit on the other hand, that one is being coded here by our phage, by your plasmid, or even your transposon. Sample for that, we have your Corynebacterium diphtheriae. That's trying to cause here of diphtheria. So, only the Corynebacterium diphtheriae which has been infected by the phage because of the toxin will be the one causing here the diphtheria. So, that diphtheria toxin is responsible here for inhibiting the protein synthesis. It try to affect your liver, your heart, and muscles. So, ganun ka-specific ang kanyang activity. Another exotoxin produced here by our Clostridium botulinum to try to cause here your flaccid paralysis among the infants. And we have also here the exfoliative toxin produced by your Staph aureus and your Strep pyogenes From the word self-exfoliative, it try to cause exfoliation or skin peeling. Skin rashes, ganun kanyang manifestation. Your exotoxin, on the other hand, to mitigate its cytotoxic activity, also require here, okay, your enzyme. Example for that, we have your hyaluronidase. Hyaluronidase try to hydrolyze your hyaluronic acid of your connective tissue that would mitigate the spread of your infection. Kaya tinawag siyang ang hyaluronidase natin as your spreading factor because again, it try to mitigate, facilitate the spread of the infection. Okay, now we go to your endotoxin. So endotoxin from the word itself is part of the bacterial cell. And so it could only be found in our gram-negative bacteria. Usually, we can find this one specifically in the outer membrane. So ulit ang ating gram-negative, di ba, may inner thin peptidoglycan layer and the outer membrane made up of your polysaccharide, polypeptide, and we have also here the LPS, lipopolysaccharide. So in this component, you could find here your endotoxin. So ito ang kaniyang basic part. Your antigenic O, polysaccharide, core polysaccharide. So ito, lipid A, the component here pertains to our endotoxin. Okay, so the effect of the endotoxin is non-specific, generalized, like I change here the blood pressure, induce fever, and we have also here the fatal condition resulting to your septic shock. So in your PowerPoint, may nakalagay dyan na table, try to tabulate the difference between your exotoxin and your endotoxin. So kindly go through na lang with that. So ito na lang. Okay, I just get some here. So again, your exotoxin is being produced here by your both gram-positive and gram-negative. Your endotoxin is only produced by gram-negative. So your exotoxin is protein by nature and your endotoxin is your lipid A. We have also here stability at the 100 degrees Celsius temperature. So it's much stable ang endotoxin. Mean to say, if try to subject this one to heat at 100 degrees Celsius, your endotoxin will not be destroyed, whereas your exotoxin will be destroyed, making that one as your heat labile, not toxin. Okay, then we have here the different mechanism here for the resistance of the host here against the entry of your pathogen for possible infection. The first one, we have here the physical barrier. So you should have an intact, healthy skin to prevent the entry of your microorganisms. Except, for example, here in the case of your Leptospira interrogans, possible for the leptospirosis, Francisella tularensis, also your treponema, where they could enter our body even if you have an intact na skin. Or you could have entry here in our skin through your bite, through the bite of the vector, mga mosquito vector, or mga ticks, ganon. So you could also enter our body. Another one, our host defense here would be the presence of your normal microbial flora would try to compete here with the pathogen for the space, nutrients, and preventing here the growth of your pathogenic bacteria. Another one, there's also responsible here for maintenance of the normal pH, like in the case of your lactobacilli in your urogenital tract. Sebaceous gland, on the other hand, try to have your fatty acid content and lysozyme, which have the antibacterial property. Okay, then we have here the cleansing mechanism. So the first one, this cremation process whereby your keratinized skin is able to peel off. Eh, pag natatanggal sila, natatanggal din ang mga bacteria natin. Another one, your tears would have here the lubricant property, also contains your secretory IgA and lysozymes which have the antibacterial property. Yung respiratory tract, meron tayong ciliated, may cilia tayo that try to filter your air that would filter the microorganism entering our respiratory tract. Okay, another one, flushing of your urine could eliminate also pathogen found in your urogenital tract. Then we have here your antimicrobial substances. So your stomach here produces hydrochloric acid that try to maintain your stomach here as a sterile site except your Helicobacter pylori, where it could resist here the acidity of your stomach. So we have also here the secretions, our body, mga body fluids natin contain secretory, IgA, and lysozymes, again, would have here the antimicrobial property. Beta-lysine is being produced by our platelets. Ang clotting process, we try to disrupt here your gram-positive peptidoglycan. Interferon would have the antiviral property. Then we have your indigenous microbial flora, help also here again in the prevention of the growth of your pathogenic bacteria. Again, they try to compete with the space, with the nutrients, so wala na sila makakain, wala nang space ang mga pathogenic bacteria, and therefore they could not grow in our body parts. They're also producing bacteriocin, which try to inhibit here the growth of the closely related species. And another one, your normal microbial flora also try to produce here metabolic byproducts, including your vitamins and essential nutrients. Okay, then we have here your phagocytosis. So we have here the outline of our phagocytosis. Start here with your chemotaxis. It's just attraction of your phagocyte towards the area where you have your pathogens. So it's being mediated here by our chemotaxins. Example for that, we have your complement component, C5a. We have also your C3a. Followed here by your diapedes is just a migration of your phagocyte from your blood going to your tissue where you have your tissue injury. Next one, we have your opsonization, the process whereby your pathogens being coated here by our opsonins like your IgG kanina with your Staphylococcus aureus na protein A, but try to block the Fc portion of the IgG. So the IgG itself could coat here your pathogen, that's opsonization, that would render your bacteria to be tasty to your phagocyte. Fourth, we have engulfment, kakainin nyo na. Then pupunta ang ating bacteria in the vacuole of your phagocyte as your phagosome. Six, we have with the fusion of your phagosome in the lysosome to form here your phagolysosome. And lastly, we have here the intracellular killing of your bacteria mediated by the granules, the enzymes present on that. Then we have here inflammation process is just our normal tissue response if you have an infection. So we have here the cardinal sign of our inflammation. So swelling, characterized by vasodilation of your blood vessels. We call this one your tumor. Redness is the increased blood flow to the area. That's your rubor. Heat would be because of the increased temperature. That's your calor. And pain we have here because of the increased pressure. Because of your tissue damage, that's your dolor. Then we have your immune response. So we have your natural or innate immunity that's non-specific. So non-specific because it would respond to whatever pathogen that enters your body. Your first line of defense includes your physical barrier, your skin, dapat intact skin and all. Another one included here, your second line of defense. We have here the cellular component of our immune system like your phagocyte or even your complement. Your adaptive, your acquired immunity on the other hand, so this one is very specific. Usually try to be activated on the second exposure. So this involves here our third line of defense. This includes your humoral mediated immunity, which involve here our antibody production with your B lymphocyte. And we have also your cell-mediated immunity, which involve here your T cell lymphocyte. So your phagocytosis, inflammation, mga immunity natin, it will be further discussed in your immunology serology na lang.